Hello everyone. Today's topic is Entomology Part 1 and we will be discussing about mosquitoes. Medical entomology can be defined as the branch of science that deals with arthropods that cause disease or that deals with vectors of organisms that can cause disease in humans. Mosquitoes in India can be divided into Anophelines and Culicines. Under Anophelines comes the Anopheles mosquito and under Culicine comes Culex, Aedes and Mansonia. Habit of mosquito. The breeding place of Anopheles mosquito is clean, small moving streams of water which includes overhead tanks, puddles of water in the forest, fountains, etc. The breeding place of Culex mosquito is dirty and polluted water which includes drainage, cesspools, septic tanks. The breeding place of Aedes mosquito is artificial collection of water, which include coconut shells, broken bottles, discarded tins, tarpaulin, flower pots, etc. The breeding place of Mansonia is the lakes or streams which contain the pistia plant. The Mansonia mosquito lays its egg on the undersurface of pistia plant and the larvae and pupae get attached to the rootlets of the pistia plant. Coming to the resting place of mosquito, Anopheles, Culex, and Aedes all are endophilic mosquitoes. That is, they rest indoor. The Mansonia mosquito is exophilic. It rests outdoor predominantly. The feeding pattern of mosquitoes. Anopheles mosquito feeds both on animals and humans. It is anthropophilic and zoophilic, whereas Culex, Aedes, and Mansonia they feed on humans, anthropophilic. Coming to the time of bite, Anopheles mosquito bites at night, dusk to early morning. Culex mosquito bites at midnight, Aedes mosquito bites at daytime, and Mansonia mosquito bites at night. The flight range of Anopheles mosquito is 3 to 5 km. Culex mosquito is 11 km, and flight range of Aedes mosquito is less than 100 meter. Anopheles mosquito is also known as sophisticated mosquito. Culex mosquito is known as nuisance mosquito and Aedes mosquito is known as tiger mosquito. Parts of the mosquito are head, thorax and abdomen with 3 pairs of legs. The parts of the mosquito head are proboscis with which the mosquito sucks the blood. Then there is a pair of palpi on either side of proboscis and then there is a pair of antennae. The life stages of the mosquito are egg, larva, pupa, and adults. The Anopheles mosquitoes rest at an angle to the surface, whereas the Culicine mosquitoes rest parallel to the surface. And the wings, Anophelines have spotted wings due to alternate dark and white bands. Culicine mosquitoes have uniform wings. The Anophelines lay egg singly. Aedes also lay eggs singly. Culex and Mansonia lay egg in clusters. Larvae of Anophila rest parallel to the water surface, whereas the larvae of Culicines they lie at an angle to the water surface with head suspended downwards. Pupae of Anophelines as well as Culicines are comma-shaped. Times, the head of the female has palpi shorter than the proboscis and the antennae is less hairy. In Culicines, the male head has palpi longer than the proboscis and bent outwards. The antennae is bushy. In Anophelines, the female head has palpi and proboscis of the same length and the palpi tip is straight. The antennae is less hairy. In Anopheline male head, the palpi and proboscis are of the same length and the palpi tip is club-shaped. The antennae is bushy. So, the males usually have bushy antenna and in Anopheline mosquito, the male and female have palpi and proboscis of the same length. Whereas in culicines, the palpi and proboscis are of different length in male and female. How will you identify Anopheles mosquito? The spotted wings. Then the palpi and proboscis of the same length. Club-shaped palpi in Anopheles male and straight-tipped palpi in Anopheles females. The Anopheles mosquito is the vector of malaria and filariasis, but not in India. Anopheles eggs are laid singly on the water surface. They are boat-shaped with lateral floats. Anopheles larvae rest parallel to the water surface. So it is a surface feeder. There is no respiratory siphon for air supply. There are palmate hairs on all the abdominal segments, and the eighth abdominal segment has respiratory spiracles. Anopheles pupae has large cephalothorax and narrow abdomen. There are paddles at the end of abdominal segments, and there is a siphon tube on the dorsal surface. Control of Anopheles mosquito. It includes anti-larval measures, anti-adult measures, and personal protection. Under anti-larval measure comes environmental methods, which include source reduction like land filling and drainage. Under chemical methods come mineral oil, Paris green or copper acetoarsenate, synthetic insecticides like temifos or abate, diflubenzuron, biolarvicidal agents like Bacillus thuringiensis and Bacillus sphericus. Under biological method include larvivorous fishes like Gambusia and Guppy. In anti-adult measures, we have indoor residual spraying, which is done with synthetic pyrethroids like alpha cypermethrin, cyflumethrine, and deltamethrin in India. The IRS is done around 50 houses of the positive case of malaria. Under genetic control comes sterile male technique and cytoplasmic incompatibility. Under personal protection comes mosquito net, which should have a hole size of 0.0475 inch. And screening can be done. Insecticide treated nets, long lasting insecticidal nets can be used. Repellents like diethyl toluamide and diethyl phenylacetamide. And also protective clothing can be used. Culex mosquito has uniform wings. The male and female can be differentiated by the bushy antenna of the male. In males, the palpi is longer than the proboscis and bent outward, whereas in females, the palpi is shorter than the proboscis. The Culex mosquito is the vector of Japanese encephalitis, West Nile fever, Bancroftian filariasis. Culex egg is laid in raft-like clusters. Each egg is cigar-shaped. The Culex larva is suspected at an angle with head downwards. It has a siphon tube which is long and slender on the eighth abdominal segment, but there is no palmate hair and no respiratory spiracles. The Culex pupa has a siphon tube which is long and tubular on the dorsal surface. Control measures for Culex mosquito, which include anti-larval measures, anti-adult measures, and personal protection. Under anti-larval measures, we have environmental method, chemical method, and biological method. In anti-adult measure, we have insecticides and genetic control. This all has been discussed in the Anopheles control. The Aedes mosquito, it can be identified with the uniform wings and the legs have alternate light and dark bands. So it is called tiger mosquito. The head of Aedes is similar to the Culex mosquito. Aedes mosquito is a vector of yellow fever, dengue fever, chikungunya, Zika, and Rift Valley fever. The Aedes egg is laid singly and it is cigar shaped or spindle shaped. The Aedes larva has siphon tube on the eighth abdominal segment which is barrel shaped, and the Aedes pupa has a siphon tube on the dorsal surface which is very short. Coming to the control of Aedes mosquito, it has anti-larval measures under which environmental method include source reduction. Dry day is observed by discarding all the artificial containers like coconut shells, unused plastic containers, discarded tins. Then there are chemical methods and biological methods similar to Anopheles and Culex. Then under the anti-adult measure comes the indoor space sprays or ISS. It is done with pyrethrum extract and kerosene in 1 is to 19 ratio using a fogging machine. Then the thermal fogging, which is done with technical malathion and diesel in 1 is to 19 ratio with a fogging machine. Then there is genetic control and also personal protection measures can be done. Mansonia mosquito is similar to other culicine mosquitoes. Mansonia mosquito is the vector of filariasis, Brugia malai, and chikungunya fever. The Mansonia egg is laid in star-shaped clusters and each egg is spindle-shaped. The Mansonia larva has a siphon tube which is triangular and chitinous to attach to the rootlets of the pistia plant on the eighth abdominal segment. The Mansonia pupa has a siphon tube which is long and narrow on the dorsal surface. The control measures for Mansonia mosquito, which include anti-larval measures, anti-adult measures, and personal protection. Under anti-larval measures, we have environmental method like removal of the pistia plant, use of herbicides. All other are similar to other mosquito control measures. With this, we come to the end of identification of mosquito. The microscopy slides of mosquito and its parts are available in my drive through this link. This link has been provided in the description. Or you can join my Telegram channel, Comet Simplified. Thank you.